Every year, billions of dollars of crops are lost due to ground ozone pollution. Hello, my name is Tristan Bissoondial, and I shall be presenting my research on the effect of ozone on the fern Ceratopteris richardii, or sea fern. Ozone is a form of oxygen gas which is made up of three oxygen atoms bonded together. It is naturally made in the stratosphere and is used to protect us from harmful UV radiation. Pollutants that formed ozone can also travel great distances by wind to Long Island, where I live. Depending on the amount, ground ozone can cause irritation of lungs, making it difficult to breathe, especially in people with asthma like myself. Many studies have shown that ozone can enter the leaves of plants. It breaks down chlorophyll, reduces photosynthesis, and slows down growth. Ozone can also increase plant sensitivity to pests. In flowering plants, ozone can reduce seed production and fruit development. Ozone can decrease pollen germination and seed production. Pollen and ovules are equivalent to sperm and egg. For my research project, I wanted to improve reproduction and development in plants. I used gametophytes of the fern Ceratopteris richardii. Ceratopteris richardii is a seedless fern. It produces spores instead of seeds. When a spore germinates and develops, it produces a haploid multicellular structure called the gametophyte, which grows as a flat two-dimensional sheet of cells, making it easier to see the effect of ozone on cell division and cell death. Since there are no studies on the effect of ozone on the gametophytes of ferns, my research question is, how does ozone affect the growth and development of gametophytes of Ceratopteris richardii? I hypothesized that as the exposure of ozone increases, there will be a decrease in growth and development of the gametophyte. I also hypothesized that the antioxidant glutathione, or GSH, will increase the plant's tolerance to ozone. And the mutant PQ45, which is resistant to herbicide, will have more tolerance to the ozone than the wild type. To test my first hypothesis, I first grew spores on a sea fern agar plates, then I exposed the spores to different amounts of ozone and measured germination. As seen in this graph, the more exposure of spores to ozone and the less germination occurs. 2 or 3 minutes of ozone dramatically reduces the germination. I did all my experiments in triplicates and did a student t-test to validate the significance of my result. Besides germination, ozone can also reduce cell division in the gametophytes. As seen, gametophytes treated with ozone had smaller protholi with fewer cells. To understand how ozone affects plants, I measured the amount of protein and chlorophyll level. I found that ozone severely reduces protein and chlorophyll level, and it is believed that protein in chlorophyll breaks down because of the high level of reactive oxygen species, or ROS. To see if ROS might be affected, I measured the activity of an enzyme that breaks down ROS. I found that ozone increases the amount of peroxidase activity. The excess of ROS may be the cause of cell death, as seen from the trypan blue stain, which measures cell death. If I pre-treated the gametophytes by adding 1 millimolar of the antioxidant glutathione for 24 hours, I could protect the gametophytes against the harmful effect of ozone. GSH can protect the gametophytes against cell death and chlorophyll breakdown, allowing for fertilization of the egg and development of the sporophyte. I wanted to see if there are plants that are naturally more resistant to ozone. If plants can break down high levels of ROS, they should be more resistant to ozone. The mutant PQ45 is resistant to the herbicide paraquat, which causes high level of ROS in plants. When I treated the PQ45 mutant or the wild type gametophytes with ozone, I found out that the PQ45 could survive higher levels of ozone than wild type. The PQ45 mutant had more chlorophyll and less cell death. In conclusion, my hypotheses were supported. Increased exposure to ozone reduces growth and development. Plants could be protected by pre-treating them with antioxidant glutathione. A mutation that makes Ceratopteris tolerant to herbicide paraquat makes it also tolerant to ozone. For my future studies, I would like to know what genes were affected in the mutant that make it tolerant to herbicide and ozone. Thank you for listening to my presentation.